staining. You can also do the tissue clear, and you can use a light sheet microscope, and you can image the 3D structure for the iris to calculate the iris volume. So this method actually with two important collaborators. So I want to highlight them. On the left is Adolf, whose group mainly did all this transplant work. So the Sarah Stanley, whose group helped develop all the iDisco 3D image technique. As you can see here, I will show you more clearly from this movie. This is the kidney, and the green is the insulin allograft after transplant to the mouse kidney. You can clearly see the 3D structure. Red is the actin from the kidney, and you can see that it's a 3D allograft structure with the transplant. You can measure this allograft by the microscopy and some software. So using this method, we can measure the LS volume, or we can say the base cell mass in 3 dimensional with different compare different treatment group. As you can see here, on the top panel is a vehicle. You can clearly see this LS group that the graph is relatively small and sparse, as you can see here. And however, when you see the Hermine plus 4, you can see this LS graph. It's much bigger and thicker, and also it's compared this is apparently it's much more expansion. This is after treat 3 months with the mini pump I just described. So using this method, we can measure all kinds of different treatment to compare the base cell volume in 3 dimensional, not just based on one histology, the staining. So here we just quantify, we can quantify the picture, the video as I just show you. So on the y-axis is human base cell volume. On the x, it's the time, the days we treatment with the different treatment. So we can see here with the Hermine treatment, it will increase about 300% of the base cell mass compared with control and X94. With Hermine plus X94, which can increase 700% of the base cell volume. However, we think this largely increased base cell volume is may not just based on just the proliferation. The reason is for in vitro, we can increase base cell proliferation about 5%. However, when we do the in vivo, somehow in vivo it's much more complicated. The base cell proliferation pretty much low, it's less than 1%. We think about 3 months treatment with this proliferation rate, it's impossible to increase so much base cell mass. So we think likely there's it's a combination mechanism including proliferation, also enhance base function, survival, and the transdifferentiation. So with this, the increase of the base cell mass, we ask, is this enough for type 1 diabetes, the therapy? I think for type 2 diabetes, the answer should be yes, because all the patient always have plenty of the base cell left. And for the type 1 diabetes, we probably need the immunomodulator therapy with the combination with this treatment. So for first part, the briefly summary that so Hermine was identified from a high-throughput screening. Third-point inhibition enables adult human base cell proliferation. Hermine alone or Hermine X94 combination dramatically expand human base cell mass in vivo. Next, we want to ask whether the effect of harming plus GLP-1 receptor on human B cell differentiation and function, will this replicating B cells work normally? So since harming or alone or harming plus GLP-1 receptor agonists can drive human B cell proliferation, we worry that this proliferation may result into some of de-differentiation and which may not have fully function. In order to test that, we treatment with the human islets in vitro with DMSO, GLP-1 harming, and GLP-1 harming combination. Surprisingly, we found that the harming alone can increase many of B cell function gene expression, such as PDS1, as you hear, also MAF-A, MAF-B, NKCP1, GLU2, even GLP-1 receptor and PCSK1. Many of the B cell function genes were induced by harming treatment. It does not cause B cell de-differentiation. Also, it's not just at RNA level. We also feel that it can increase the B cell function gene expression at a protein level as well, as shown here, PDS1, MAF-A, and NKCP1. And this combination also actually can increase beta insulin secretion, but does not change the insulin content. So this is not only happening in normal B cells, it's also happening in type 2 diabetes islets. So this will potentially for future type 2 diabetes treatment. So we also ask whether this combination can rescue the diabetes. So what we did is we use the same condition. But we use STZ to induce the immunodeficient mice into the diabetic mice, and then we transplant human islets. We use a marginal transplant, and we treatment with this mice with the mini pump, either vehicle, extended fall, harming, or combination. I want to point out here, for treatment with harming, we only use 1/10 of the normal harming amount. The reason is we want to see the synergistic effect. If we use normal harming, if we use the normal harm dose, we will reduce glucose level rapidly in 1 week. That's why we want to see the combination effect. We decide, okay, let's cut the harming, the 1/10 dose, the combination, everything is 1/10 dose. In that way, we want to see whether we can see the effects together. So as you can see the data here, we can see that harming and extended fall can have the synergistic effect. After 1 week treatment, will reduce the blood glucose to the normal glucose level. We follow up for about 3 months, and we can see that it's staying in the normal glucose level for the 3 months. We also found that this harming extended fall treatment can increase the plasma insulin level as well. Since we found harming can drive human B cell function gene expression, we want to see the mechanism. And we want to see whether all of the DARPA inhibitor which can drive human B cell proliferation have similar effects. So we treat the human islets with all of the DARPA inhibitor commercially available, so which include harming, 22C, 5-IT, GNF, IND, lucitin, CC401. Surprisingly, we found that only harming, 22C, 5-IT, 3 of the DARPA inhibitors can drive B cell function gene, as shown here, PDS1, MAF-A, NKCP1. However, all the other DARPA inhibitors, although all of them can drive equally for the B cell proliferation, based on publication, actually, I also test myself. Indeed, all of them can drive human B cell proliferation. Sorry, I just recover from this cold. So it's really interesting that it means that likely B cell function induction and base cell proliferation, it's different targets. It has different targets. So we also did a single cell RNA-seq and try to compare what cell types have this induction. So as you can see here, this group actually is defined as base cell group population. You can see here the DMSO and the Hermine treatment. For example, for PDS1, you can clearly see that that Hermine treatment will increase the PDS1 gene expression mainly in the base cell group. Also the MFA in the base cell group, you can clearly see based on color code so change. And also in case PO1 and the GLU2. The only one small population maybe it's second alpha. We define this as second. It's one of the alpha group, but it's a second alpha. We think there's a possible that this second alpha after treatment can reprogram into base cells. We have a paper published, but I will not talk about this group today. So just mention that this group also, increase this alpha cell, but with this treatment, you can see there's a base cell signature gene expression here as well. So next we want to see whether DRG1A and 1B involved in this base cell function gene expression. So we have the verse, I think we published GCF for this verse that when we made one verse can silence both DRG1A and DRG1B, we can see base cell proliferation. So we use the same verse, we want to see whether we can see that can increase base cell function gene expression. As you can see here, the verse work very well. You can silence DRG1A and DRG1B here. And when we transduct this virus to the human atlas, as you can see here, we have 4 conditions. We have control alone, control plus Hermine, and silence DRG1A and 1B, silence DRG1A and 1B plus Hermine. As you can see here, Hermine plus control can increase base cell function gene expression. However, when we silence DRG1A and 1B, we don't see any base cell function gene expression change. When we add back Hermine to the even DRG1A and 1B silence, so we can see the base cell function gene expression again. So it suggests that this base cell function gene does not request DRG1A and DRG1B. It's independent DRG1A and 1B. We also test many other pathways which potentially involve into the either base cell proliferation based on our earlier publication. And also we know that based on our canal scan, which I will not show today, that it's Hermine also can inhibit this different kinase pathway. From our canal scan, we know that Hermine also can inhibit whole DRG family, including DRG1A, DRG1B, DRG2, DRG3, DRG4. Hermine also can inhibit the CLK family, CLK1, 2, 4. So we made a verse and can silence one verse can silence whole family, and then we test in human atlas. As you can see here, compared with control, we only see maybe one gene MFA. We can indeed see some of induction, but not like Hermine whole panel of the base cell function gene change. Only one gene change. So it suggests DRG1A, 1B, this family may have some member may be involved, but not the key thing. And we also silence the CLK family. We don't see obvious change between the control group compared with the control group. And we also test the NFAS pathway. The reason is we test the NFAS pathway because from our Nature Medicine paper, we know that NFAS involved into the base cell proliferation. That's why we overexpress all this NFAS member constitutively. And then we check base cell function gene expression. We don't see them increase at all. However, we do see that opposite results will reduce some of the base cell function gene expression as shown here. For example, MFA, PDS1, and NKCP1. And we also check a dream complex pathway. I published a paper on GCI paper that paper showed that dream complex involved into the base cell proliferation induced by DRG1 inhibitors. So we know that E2F4, E2F5, and P130 and P107 is a key member for the dream complex. When we silence this member, we can see proliferation in human islets. However, when we use same verse silence in the human islets, we don't see any obvious change for base cell function gene expression. So it suggests that the dream complex involved into the base cell proliferation induction, but does not involve in this base cell function gene expression. So in order to embarrass, they find the protein interact with the hermine, which can drive human base cell function gene expression. We decided to use an unbiased way to screen the compound. What we did is we have a chemical group collaborate with us at Mount Sinai. So they have modified this hermine, and they added the different linker to this hermine. In this way, the modified hermine still can drive human base cell proliferation, still can drive human base cell function, which we test in human islets. But this linker can binding with this magnetic beads. So with this modification, so we can interact with the islet protein with this compound. Then we can use another compound, which is a newer generation of a very potent hermine, the 22C, and we can competing with this interaction and competing this binding proteins. So in this way, then we can also mass spec to test sequence all these proteins. So based on binding affinity, we can rank those candidates. So we recognize about 25 of new proteins. As you show here, dark one was one of the list, which is validate this binding approach. It means this approach works well. And we made a verse, all these 25 verse the proteins, we overexpress them and silence them because I'm running the verse call. So I took advantage of this, this way we can make any verse we want. So we can silence, overexpress them. So we made this verse, we overexpress and silence this verse in human islets. Then we test the base cell function gene. Only one of them we can see when we silence this, we can see some of base cell function gene expression like a hermine treatment, which is PRKAR1A upon here. Although it's not a high rank, but actually it's have the very amazing phenotype. So let's see what's a PRKAR1A. So actually PRKAR1A, it's a PKA regulatory subunits. So for PKA, there's 4 subunits. There are 2 of them catalytic subunits, which is the PRKACA and the PRKACB. There's another 2 is actually PRKAR1A, 1B, or PRKAR2A, 2B. In base cells, PRKAR1A is the most abundant one. So then when in the basal level, PKA staying inactive, this regulatory the PRK subunit will binding with the PKA catalytic subunits. With the second MP increase, it will be able to binding with the PRKAR1A or PRKAR2A, which will disassociate the PKA catalytic subunit from this the complex, which will active PKA activities and increase the PK activities, which can phosphorylate downstream transcription factors, such as the typical ones, CREB, and many other ATF family. So we think if hermine indeed can increase binding with PRKAR1A, we assume it should be able to increase PK activities. So we did Western, we test whether can increase downstream the PKA, downstream transcription factor phosphorylation. So what we did here, we treatment both base cell line, also the human islets with harming. And also we use a PKA activator fuction. We also have another inhibitor. We know that can drive human base cell proliferation, but it cannot increase base cell function gene expression. As you can see here, indeed, we can see that harming can increase phospho-crab in both R71 base cell line and human islets. Fuction also, we know that as PKA activators, it also can increase both. Although fuction is stronger than harming. We also did the immunostaining, and we can clearly see this happening in base cell, showing that insulin positive and phospho-crab positive base cells. This suggests indeed harming can increase PKA activities, but indeed does not. Next, we ask if this PKA indeed, if harming can increase base cell, can increase PKA activities. So the PKA should be able to increase base cell function gene expression as well. So we choose several of the PKA activators, including fuction, ACPT, second MP mimetics, also 6-benzene second MP, and dibutyl second MP. This is typical of the PKA activators. Then we treat the human islets. Then we measure base cell function gene expression. As you can see here, clearly all of these PKA activator can increase base cell function gene expression as shown here. Similar like harming treatment at RNA level, and also we measure the protein level based on staining. You can clearly see compared with the control, there's a much more MFA expression with the harming, also the fuction, but not with the IND. And for PDS1, you can clearly see there's more of the PDS1 gene expression with harming and fuction. And even more, that we also think if that's true, when we use PK inhibitor, we should be able to block all the effect induced by harming. So we use a typical classical PK inhibitor, which is H89. We treatment with DMSO harming in the H89 and harming plus H89. As shown data here, harming can increase PDS1 MFA gene expression, and but not another DARK1 inhibitor India, which we already know. And PK inhibitor does not change PDS1 or MFA gene expression. However, when we have harming plus H89, it indeed will reduce harming-induced PD-1 gene expression, also reduce harming-induced MFA gene expression. So next, we want to test genetically test this pathway. So we made the constitutive active PKA verse, and we also made the silence the verse to silence PRKLR1A, as I mentioned earlier. And as you can see here, when we overactivate PKA, we do see there's more of the PDS1 gene expression, more of the PK MFA gene expression. It's comparable with harming treatment alone. And with combination, we can see even more of this induction. And also, we can clearly see when we silence PRKLR1A, we can see clearly there's more of the PDS1 and more of the MFA gene expression. With combination, we can see even more of this induction of the PDS1 and MFA gene expression. So this suggests indeed maybe harming induced base cell function gene by interact with PRKLR1A somehow can increase PKA activities and increase base cell function gene expression. So I was just summary briefly about this the pathway. We think DARK1A, some of DARK1A can have dual function. So most of DARK1A can have the can drive base cell proliferation, but a few of DARK1 inhibitor can have dual function, increase base cell proliferation through inhibit DARK1A, and also can increase the PKA pathway and drive base cell differentiation function gene expression. Next, I want to take the last few minutes to talk about safety. So since safety is always the most important for the future clinical, those kind of development, we know that harming DRPA is ubiquitous present in many tissues. So will harming alone or with Claudin-4 cause proliferation of other tissues? We'll have to address this before we can move to next stage. So in order to address those questions, so when we do the 3 months treatment with harming Claudin-4, we also not only collect the kidney, we also collect many other organ tissues such as liver tissue, spleen tissue, intestine tissue, lung, heart, and kidney, and pancreas. Then we did a Cas7 staining on these tissues. We don't really see obvious difference between each of the treatment. The only thing we see some more proliferation is like Claudin-4 in the lung. However, we don't see the combination. There's more proliferation for lung as well. So overall, it's no unwanted proliferation in other organs. We also did the histology staining to see whether there was an unwanted structure, and this is the same tissue we collect when we do the 3 months treatment. So we also do not find abnormal histology in other organs as well. Because of this very encouraging data, we decided, okay, it's time to move on to the next stage. We decided to go ahead to do the human phase 1 clinical trial with the pure harming. So this clinical trial was conducted by the Dr. Marot from the Mount Sinai, and also we produced this GMP quality harming, and we got FDA approval, and we got Arabic approval. We got money from NIH. Actually, NIH very generous, and very much support this kind of clinical trial. We also registered the clinical trial, and this is the pure harming formula. It's 100 milligram capsule, and we designed this clinical trial is dose escalation way, which means that you can give the participation from low to high dose until they cannot suffer. Then you can stop at the highest dose. We suppose we design from 100 the highest possible 1200, but one patient still have some side effect at 500 milligrams. So we highest possible dose, so we stop at 500. So we already finished this clinical phase 1 clinical trial. Overall, there's about 34 people signed the contract, and there are 25 people received the harming, and here's the different dose, and also these 24 people also finish follow up and finish all the assessment. And overall, there's no severe side effects, no deaths, and the only side effect is nausea and vomiting when we use a high dose. Also, we did the dose limiting toxicity, so no any bad side effects, and there are also no visual hallucination, and only nausea and vomiting at the higher dose. So for the prediction for the future, human B-cell regenerative drug therapy is clearly possible. It will be effective in tens of millions of people we tap to initially, then move to type 1D when immunological challenges are met. It will be much cheaper and far more scalable than B-cell replacement strategies. It will be at least as safe as other replacement therapies in current or future use. A brief 3 months treatment may be sufficient. Also, there are some of the current challenges. We can move to the next generation of harming to human trials. The reason is, although we did a harming treatment, clinical trial, but this is nature product. We could not patent that. That's why if we don't have patent, pharmaceutical company they don't buy. That's why we developed the newer generation. We have a collaborative group have produced like 1,000 of the newer generation of the harming, which we have patent. That's why we can listen to the company. So, and it's moving well for their part. So we're also managing the autoimmunity for type 1 diabetes. And also continue synthesis and explore and develop and next generation DRC inhibitors. Last, so since this is multiple group, it's a big project, involve many people. I cannot list them all. So for the Monsana DOMI part, Andy and I are the co-leading this whole project for the most of the discovery and the biology mechanism. We also have the drug discovery and the chemical group leading by Dr. Davido and Puno. And also we have the clinical trial group, which is leading by the Dr. Murray group, and also Dr. Adolf, who was in the Monsana DOMI member, now moved to the City Hope. And also thanks all the funding agency, also the donor and the families. And thanks for your attention. Yeah, I will take some questions.
Okay, great. Thank you so much, Dr. Wang, for this like a really insightful and inspiring talk. I want to give you a special thanks since you just
Thank you.
still recovering from the cold.
Yeah, I'm still on the coffee.
Yeah. I just feel like all those like research you have done start from like unbear screening until the clinical trial is really exciting.
Right.
Yeah. So with that, I think we will move to the panel discussion part. So we are delicate to have three outstanding panelists join us today. So we will start with the questions with the panelists first. After that, we will open the floor to the audience questions. So first, I will introduce Dr. Wenhong Li from UT Southwestern to share his comments or suggestions or questions. Please, Dr. Li.
Hi, Peng. Nice talk.
Yeah.
And
Yes, thank you.
Congratulations on the initiation of the clinical trial. That's always what we are aiming for by the end of the day. So that's very exciting. Um, I just have a few quick questions. The first one, in the beginning, you mentioned from the structural modeling or maybe X-ray structure, harming was proposed to bind to the ATP site of the DERK kinase. What's is anything unique about the ATP bind site in this DERK kinase that may guide you to design more specific binder for the DERK kinase and then try to reduce non-specific binding to other kinase? That's my first question. The second one is when you assay this beta cell proliferation in human islets, did you notice any stimulation effect on alpha cell proliferation from harming?
Okay, that's a great question. And for first question, I don't know exactly for the structure parts, but I do know that from several different DRC inhibitors, they do have a small difference for the ATP pocket. So we do see a lot of the kinase scan from different DRC inhibitor. I don't know exactly, why they are different, but I think, from the structure, it's very hard to say, but from chemistry, the P group, they always show even the minimum of the amino acid change, they can see the structure change. But for my set, to be honest, I could not clearly tell you how the, specific difference for the ATP pockets. But I know there's a difference, but I can know how to say that. I'm not chemistry people. We have a group, they can tell, doing the structure, they are working for us. So, and we do have some other experience is, I think they separate this like a type 1 kinase group, type 2 kinase group. I think this they call the ATP pocket base. I believe I remember clearly, if I remember right, they call that type 2 kinase inhibitor. They also trying to develop some not based on ATP pocket DRC inhibitors, they call type 1. I think they based on the mainly based on the binding structure, the compound can hook there, not based on ATP. Based on ATP it's relatively there are a lot of limits because many compound have ATP pocket easily have off-target effects. That's why we have some new compound which is not based on ATP pocket. It's based on mainly dopamine inhibitor structure. So that's for that. I don't know very details how to answer about that. So that's for first question. Your second question, you ask whether this affect alpha cells, right, for proliferation. So my answer is indeed we do find harming can driven alpha cell, somatostatin cell, B cell proliferation as well. However, that's why I make one study very much exciting. It was a combination with the GLP-1 receptor, and Courtney found that when harming at a very low dose, can with the combination with the GLP-1. So because harming can drive B cell proliferation high when it's like a 10 micromolar. When Courtney use a low dose only use 1 micromolar, then when he combination with the GLP-1, it can drive similar level of the B cell proliferation like a 10 micromolar. However, in this dose, because the alpha cell does not have the GLP-1 receptor, she does not see the proliferation from the alpha cells. In her case, she only sees that proliferation in B cells.
Okay, great. Well, given how popular GLP-1 agonist is nowadays, that
Right.
just increase the chance of
Right.
to have harming to be more selective against the beta cells.
Right.
Thank you.
Yeah.
Okay, thanks, Dr. Li, for the question. So next, Dr. Wang, Li Heng, please.
Hi, Peng. Great talk.
Yeah.
And
Thank you.
congratulations that I saw like Harming finally moved to the clinic trial. So and say congratulations to Andy too. So my first question is actually, you mentioned that block, so actually inhibit DYRK1A by replace the ATP like pocket. So but later on you say like it's not only increase beta cell proliferation, but also help with the beta cell differentiation by activating PKA. So I just wonder the effect of harming on PKA pathway, is that direct or an indirect effect? Have you guys ever?
Yeah, I know your point. It's a very good
that.
question. Actually, that's a very, very great question. Actually, I will tell you directly for now. So we found the harming can interact PRK-L1A, right? We found PK because we found this pull down interaction. We assume that harming supposed can interact PRK-L1A. That's why we can get this pull down. However, so when we do some verification, so I have several different ways to verify the. Actually, I can show the group some data. I have two different ways to verify this interaction. So one way is I don't know whether you people understand the thermal shift. So these are. We find one way we can measure the interaction directly or not directly by the thermal shift. The principle is that if it's directly interact, the compound or any of these kind of elements can protect the protein at a high temperature, denature temperature. However, if not interact, you'll not be able to protect this the protein denature. So we had a collaboration group which is amazing in his field, and they help us to make this thermal shift. So what they did is, for example, PRK-AR1A here, and so they have the positive control, which is the second AMP mimetics, which can suppose interact PRK-AR1A. So you can see here at the different temperature. So this with the vehicle, you can see all the PRK-AR1A peak is here. It's degraded with the higher temperature. However, when they add the second AMP, it can protect this protein degradation. It's a peak shift to here. This is called thermal shift to 70 degrees. So use the same method. They did interaction with harming for PRK-AR1A. As you can see here, harming the major peak still at this like a 42 degree. It's only a small peak moved here, but not majority like here. So based on this parameter, we think likely there's something else in between PRK-AR1A, and it must be a big complex here. So it's not directly binding with PRK-AR1A. Somehow we put down this PRK-AR1A through a complex. Actually, I did this thermal shift a different way myself that we did based on Western. I can clearly see that, Hermine can protect drug A very well, and I was tracking very well. And the PRK-AR1A was protect second AMP. I do not see indeed Hermine protect the PRK-AR1A very well. Indeed, you have very good questions, but indeed, we think it's not directly interact. It's indirectly put down. So yeah.
Yeah, and also based on like your findings, I just speculate. So compared to like the use harming like alone, so it can be like to combine like some like beta cell proliferation drug and plus some like phosphocholine or PKA agonist to help to to do the same thing as harming. And with that, you may have more strategy like to find good drugs to
So
yeah.
yeah, I mean, so that's indeed we have a paper. I think that we currently did in the Science Translational paper. She did combine a lot of PKA. I did not show. I just don't have time to show all the data. I just pick some major one. Indeed, in her paper, she already showed that harming plus some of the PKs as the activator. She does see more of the preparation as well. So not just basic function. Somehow it's affect both ways. So yeah.
Okay, so my last question is because we are aiming like to use harming like to help to preserve the beta cell function in type 1 diabetes, but
Yeah.
if the reason is coming from the immune attraction, the microenvironment is not changed. If you only increase the beta cell proliferation,
Right.
how can that cure type 1 diabetes? I just wonder, does harming also have some beneficial effect in terms of on the like immune
Yeah.
environment to reduce the
That
inflammation or those things?
That's a very good point too. So actually, I think you probably, I mean, other people don't know. The Brian, right, from the Adolf group, he did find that harming have that function. So involved into the immunoprotection for the B cell attack. I think he prepared his paper. It's yeah. So the other group, he's the one doing this response reaction with harming. So and also the 24. So yes.
Okay, glad to hear that.
Yeah,
Congratulations.
thank you. Yeah. I think his paper will come out soon. So yeah.
That's exciting. Thanks, Li Heng.
Thank you. Yeah.
The next, let's hear from Dr. Meng and Zhou Xian from Zhejiang University.
All right. Okay. Great. Peng, this great talk. Congratulations on
Thank you.
a clinical trial. It's very, very exciting. It's actually the first compound I've ever heard that can induce human beta cell proliferation. So actually, I have a couple of questions. The first one is, I think the first one is related to the Dr. Li Wenhong, and he talked about the source And also there's a question in the chat box. So whether Helming can have any the source of cell proliferation, right? You say beta cell can be proliferate, but it's in the field there's always a debate whether mature beta cell can be proliferated, especially in the adults, right? And people
Right.
think maybe other cells like alpha cell, dedifferentiation, or other ductal cell and the endothelial cell maybe can also um, proliferate or transdifferentiate into beta cell. So
Right.
I'm wondering whether Hamming can target other cells instead only mature beta cell to proliferate. So have
Yes,
you?
yes. Yeah. I mean, when couple of years ago, yeah, when we published Nature Medicine paper, yeah, we do check other cell type proliferation. I do see if at a normal dose, like a full powerful dose, we do see harming increase not only beta cells. It's also increased alpha cells, somatostatin cells. I think also some of the dark cell as well. So
Mm-mm.
yeah, we do see
So
it's
this is
not unique. Yeah.
Yeah, this is very important because in type 1 diabetes,
Mhm.
the remaining beta cell is not that many. So I guess other cell
Right.
type, if can be target, that would be
Right.
better
Yeah.
for the therapy. Um,
Yeah.
so the second question is because I think your target DYRK1 is
Dirk.
actually
Yeah. Dirk.
ubiquitously expressed in almost all the cells. I'm
Right.
wondering why when you do the treatment, it only target the pancreas and or only target the insulin secreted cells. What's the select? Why is so is what's the where's the
I
selectivity come from? Or maybe
yeah.
because of the compound chemistry property that lead the compound directly into the pancreas, or this other because
So yeah.
yeah,
So I know
yeah,
you're saying why only
pancreas
packet
beta
only you're preferring the base cells, not other cell types.
other tissues maybe.
Yeah, I think for the base cell proliferation is very unique, different. I mean, a lot of things still unknown, and
Yeah,
yeah. So I think somehow I did the base cell proliferation for so many years. I just feel like there are, even we found some proliferation. There are a lot of mechanisms we don't know. I mean, a lot of times we found this compound. Uh, it's very weird when you treat with some tissue with some, naturally easy proliferation, that kind of property, you don't see effect at all, especially for like cell lines. When I use Herming, use some base cell line, which can proliferation. Somehow it's inhibit the proliferation. However, the base cell, I think this is a different mechanism. But probably that's one of related what I reported that the dream complex. Not all these kind of cell have dream complex. Only some of us have this dream complex. I think base cells are one of the cell types that have dream complex, which majorly limited all this cell cycle gene expression with this complex. I think likely, I'm just guessing, I don't have very direct evidence for that. Likely because maybe the base cell have this complex harming itself very strongly destroys this dream complex. So it also depends on how much is the proliferate really on this complex. So I think a
yeah.
base cell likely very really on this. I think it's just based on my own data, I'm guessing,
Yeah,
but I
I
don't,
guess cell
yeah,
complex. Yeah, cell complex. The complex probably is one of the determining because
right,
the other thing because in the end you actually try to modify the Hamming so that you have the pattern to license out to the company.
right,
But I think when you really modify the compound property chemistry will be also changed. In that
right,
case, maybe the distribution of the drug to the different tissue in the body will also change. That's also
right,
what happened to our. We also did some kind of drug screen, and
uh-huh,
we also experienced when we modify the compound, the distribution of the compound in the body is totally changed. So
right,
I'm not sure
right,
whether that
right.
maybe in the end Hami is the best. I'm not sure. Yeah. All right.
Yeah. So, yeah,
The chemistry itself probably is very important for the
right.
distribution.
We have tested some the chemistry group. They are amazing. They made 1,000 compounds for us. So we tested many of them. So
So
we also
I have
have
a last question. So because we know that the insulin secretion, the dynamic first phase, second phase insulin secretion is very important, especially for the normal fluctuation of the glucose control. I
Right.
noticed when you have the Hamming treatment, both on the low glucose concentration and high glucose concentration, the insulin secretion all increase.
Right.
Have you ever measured the dynamic of insulin secretion instead only the secretion?
That's a very good question too. So actually, no, actually for the short term, the in vitro, we only for now only did the like a static the glucose insulation. But the dynamic one actually, I have a recent R1 I submit that that part I did proposal to do the dynamic, but right now we haven't done that yet. No. Yeah.
Yeah, because that's particularly important for the fluctuation of the glucose control.
Yeah. We do notice that when I first time submit our other review also asked same question. So actually, we point out this as well. So that's why, yeah. So
yeah, good luck with your R1 application. All right, that's
yeah,
all for my question. Yeah, thank you. Yeah.
Yeah, so thank you so much, Zhiqian, for the really nice questions. And so with next, we will move to the question from audience. We do have a lot of questions in the chat right now, actually. So the first question is from Shiqi. Do you want to ask question directly? Okay, so I will just read the question out. So Hamming promotes both the beta cell proliferation and maturation. However, the matured beta cell have less potential for peripheral, as shown in the natal and adult proliferation rate. My question is whether an order of beta cell reaction were observed during the Hamming induction, such as the first proliferation followed by the maturation?
well, I mean that's a good question, but I mean we did not test because the proliferation overall is still low, very hard to trace which cell is proliferated. All we do that we based on the staining KC7 BRDO. When we detect the cells are already dead. We could not measure whether mature or not. But we can. I would say for early stage we can. I mean I remember I did some of the staining costing with the PDS1. Indeed, for some of the KC7 marker cells, there was a relative lower of the PDS1 gene expression, but that's just like a very short time after treatment, like 3 days. But I believe after long-term treatment, those cells will be once stop the reaction from the proliferation signal, it will eventually become maturation as well. I believe that proliferation should be start first for the specific proliferation cells, then eventually become maturation. But overall, what we see the maturation, also the base cell function, it's from a majority of the base cells, may not just reflect the cell from the proliferation cells. It's generally like a whole body of the human islets. Yeah.
Okay.
Yeah.
Yeah, I guess you have answered the question pretty well. Okay. So the next question from the chat is Chu Ping from University of Hong Kong. Are you still here? Do you want to ask question directly?
It's okay. I'm reading the question. So,
okay. Yeah.
Proven glucose. Yeah, I guess I'm pretty sure there's a newly the proliferation cells. I believe there's a reprogram happened. The reason is we have another project that we're ongoing. Actually, I did not report here because of the time problem. So we do see that we do think there's a other cell type. For example, I just use a single cell RNA-seq. We do see that alpha cell population based on the, can somehow reprogram in base cells. We are actually developing some new way to verify this. We use a stem cell. We knock out, we add some trace, because we try to trace that directly in human base cells. I mean, human alpha cells. Unfortunately, there's no promoter that can only express in alpha cell. This kind of tracing the promoter available. So we have struggled for several years. Now we decided move on using the stem cell developed the base cell alpha cells. So right now we're doing the knock in some specific marker under the alpha cell promoter. Then we using this stem cell, we can develop into alpha cell. Then we treat with hermine and other. So you can compare whether this reprogram indeed happen or not. So we don't have exact data yet, but it's ongoing. So yeah.
Okay, great question.
Yeah.
Then a question from Fu Biaoshi. Do you want to ask questions directly about the PK activation in the adipose tissue?
Actually, I could not answer this question because I'm really not know much
Okay.
about adipose tissue. So I'm
How
so sorry.
how about the mouse body weight? Do you see any body weight difference when you treat the mice with
No,
some
body
cells
weight
that-
actually it's a similar. Yeah.
Okay,
Not really
okay.
change much. Yeah.
Okay. And then we have a question from Hui, which
Mhm.
about the GLP-1 agonist
Mhm.
combined with humming.
Uh-huh.
So the question is any effect of humming and humming plus GLP-1 RA on the preserving function of beta cell mass or glucose regulation in type 2 diabetes preclinical mouse model other than human transplantation model. Any other models? This is the first question. Second
So
is
yeah.
Please go ahead, answer.
We right now only did the marginal the type 1 diabetes model. We did not do the type 2. We have grant pending which will do the type 2, but we haven't started to do that yet. So we waiting for the money is crazy. So time. So yeah.
Yeah. That
So
makes a lot of sense.
yeah. So we only do the casing right now. So with
Yeah.
limited money. So yeah. Does hermine show any long-lasting glucose lowering effect after drug withdrawal?
Yeah.
Let me think. We actually we did not do that. I don't think we withdraw the hermine. I don't. We collect grafts. So we stop the hermine treatment. We continue feed the graft because we use a million pound can constitutively feed the mass, the hermine every second.
Yeah.
So we really not. It's not like an IP injection. Those things. So yeah. No, we did not know that. So yeah. Because we can save time even with the mini pump. You don't have the technician to check every day. Once you bury the pump under the scan, you don't need to do that. You only need to measure glucose. That's all. So yeah.
How long the pump you guys are using can
They have a
last?
different size of pump. We use the one kind. We replace the pump every month. You can sustain like a month. So yeah, we can replace that every month. So yeah.
Yeah. This is actually a great question. I like the question because people know the GLP-1, right? If you withdraw GLP-1, just bump up the body weight crazily. So
yeah.
Okay. So another question. Next question is from Kenny Wang. Yes, about the GLP-1.
What's the mechanism?
Yes.
It's a great question. But I mean, right now for that paper, we only found that actually GLP-1 somehow can increase the cyclin A, the nuclear translocation, also the I think harming itself can increase cyclin A like RNA level, but somehow this RNA not into this cyclin A not really expressing in the nuclear. I think GLP-1 somehow make more of the protein level cyclin A expressing in nuclear, but that's just based on like a downstream effect. I really don't know exactly the upstream of the mechanism. We think PK involved in this. So it's also another R1 we just get, we applied, but we did not get a very good score. We have to resend about the how synergistic this mechanism. So yes.
That's a lot of directions. For this question, I guess Dr. Zhuo Xianmeng may have some opinion because he just published a paper about the GLP-1 regulation, the buff 16C, if I remember correctly, about the beta cell function. I don't know. Hey, Zhuo Xian, are you still here? Do you want to add any insights or comments to this question? Okay, I guess he's busy. He's another meeting now. Okay. Yeah. So there's a couple mechanism. I think it's probably worthy to explore more. We do have one question from iPhone.
My question I just be mentioned by Zhuo Xian, and I just see that like the JAK1A has very low expression specificity to the organs, and why it only increase the regeneration in the beta cell, but not other cells, especially like the other organs, including the liver or other like the lungs, like that.
Yeah. I think Dr. Wang answered that question because the dream complex, right? It's
right. We did. Other people are.
Yeah. Okay. Thanks, Jing.
Thank you.
So next, does harming alter allied inflammation, such as macrophage infiltration?
We have people work on this immune response. It has some effect, but I could not tell because they are still ongoing the project. So yes. Yeah,
Okay.
paper not published out. They are preparing paper right now. So yeah.
Nice. That's really productive. Then a question from Dr. Jie Qiao. Do you want to ask a question directly? Okay, so Dr. Qiao's questions related to the patients. Yeah. Connie syndrome, they have the PRKR1A mutation. Is that possible to be treated with harming?
Well, it's a kind of sensitive question. I really don't know. Cannot give you instruction for that. So yeah.
Okay.
So
maybe you guys can talk more later to see if there's a possibility to do a cooperation. I saw we have raised hands. Do you want to ask questions?
Oh yeah, sure. Thank you, Dr. Peng, for a really interesting talk. And my question also related to the Parker 1A, because I do have some experience of, about this gene. So if we knock out this gene in beta cell, it will mimicking a insulin secretagogue effect, which the beta cell just dump out all the insulin. So have you ever observed like any effect like that, or you've shown a really nice graph with the identity gene, but did you ever do like a Jesus test?
Not yet. That's a good question. I do read some paper about the knockout mice increase in insulin secretion. It's in the proposal, but we haven't done this yet. The method. So it's that's a good question, but we haven't done yet. But it will do so. Yeah.
Okay, thank you.
Yeah.
Okay, great.
Yeah.
Shumi, do you have any questions? Or comments? Or share?
Yeah, hi, Dr. Wang. Very exciting story, and I'm here to learn something about this,
Thank you.
beta cell thing, because just I'm curious about your application to type 2 diabetes patients, because in my impression, type 2 diabetes could have different stages. For example, at the beginning, the beta cell compensation stage, then insulin resistance, and finally the exhaustion of beta cells. So it looks like this application may happen in the very late stage. Is that your intention to apply this during that stage, or how this drug can improve type 2 diabetes?
So I think for type 2, so first thing, the at beginning of when type 2 happens, more like a function, like a defect, right? So I mean there are type 2 not just the function defect, it also there's eventually will cause base cell mass reduce as well, right? So I think that somehow the harm we know that it can increase the base cell like a function, and also increase, also it's not just the base cell function gene marker. We do see that insulin secretion, those things, and also I think, not just increase the base function, increase the base cell mass. It will make up all this, eventually lost the base cell function for the type 2. I think that's definitely will help for the therapy in type 2. So yeah, but we really need more experiment on that. So right now, so.
Yeah, so it looks like still have some challenge to for the application. And another thing is you explored the effect on beta cell regeneration and maturation. Have you ever looked at its impact on cell death?
We do actually. We have two different science translational paper. Another in vivo science translational paper, that paper indeed explore of the cell death. So we do find that harming can increase the EGFR or EGF. I think somehow can block cells proliferation. Actually, in my own hands, I do notice there are several cell deaths. Downstream like a TXNIP, those genes largely reduced after hormone treatment, which all relate to cell death. It somehow blocks cell death, enhance cell survival. Yeah, so we have some study about that too. So yeah.
It looks like different aspects to increase beta mass through different mechanisms. Yeah.
Right. Because it's a hit different pathway. Generally, most compound only hit one like a major pathway. You can only see one phenotype. Somehow this one, it's very amazing. Hits several different pathway, which is beneficial for all of these therapies. So yeah.
Very exciting, especially like clinical application.
Actually, when we do those things, when the company, outside company, before they bought those things, they have to use a third party, the place verify all the data. They're exactly everything they can verify that. So that's very a lot of stuff. So so that's very happy thing for that. They have a third party verify all the data. So yeah.
Yeah, congratulations. I don't
Thank
have
you.
any further questions.
Yeah.
Lihang, do you have more questions?
No, I don't want to. Yeah, if it's I don't want to say bye, goodbye to everyone.
Okay. We don't have one more question. Kim in the chat, Jianhua. Talk about, could you please comment on
menin inhibition? Oh yeah, the menin inhibition.
Okay.
That inhibitor we do test as well. It's so the menin inhibitor is very toxic, I would say. It's just like a crazy toxic. When you treat it's a manual base cell, even now die, if you stay the PDS1, MAFA, those base cell function gene, it's all gone. It's like nothing there. I test several different menin inhibitors, including the one earlier. Also, they trying to do some clinical trial, but I think they failed. So this is too toxic one. It's a kind of compare with Garmin, I would say. Not because I do harm it, because indeed we did side by side compare for those things. So it's very toxic. So somehow. I don't know. I think it's also the off-target effects make this very sick, make cells sick. So yeah.
So I do have like one question. I was wondering, so during the development, right, the DAC1A expression was like increased along the development, right? I can understand partially understand the physiological meaningful for that, because you don't want like people all the animal secretion too much insulin, because that would be really dangerous, right? But is there any side effects, or if people use harming to treat it, is that like gonna lead to like uncontrolled or too much beta cell proliferation? Is that also will cause some?
You see that we do the animal model for 3-month treatment. The mass actually even increase sevenfold of the base cell mass, but it's the glucose level staying around like 200. It's not like can respond very well to the the everyday the full change everything. I think it maintains this pretty well. It seems like this based on I don't think we need to worry about that based on, but this is all based on the mouse data, right? So yeah.
So can you give like any a little bit more background about like how the DAC1 gene is being regulated during development? What's upstream regulator for the DAC1A?
Oh, upstream DAC1A.
Yeah.
Let me think. There are a couple of them, but some MST1 something LAST. I don't couple of kinases. It's all regulate DAC1A, but I think all of this we go downstream of DAC1A. We did not really go up of DAC1A for our study. Yeah.
That's probably makes sense. If there's a lot of regulator, you probably just
target. It's very complicated. Yeah, exactly. Especially for kinase, there are so many kinase regulations. So yeah.
I see.
So that's why every time when I'm hearing this story, I feel Harmony is so magic. It's a magic drug to preserve beta cell function.
Yeah. I feel like that's 20 years of your work is really exciting. That's basically, I just feel like every scientist's probably dream will be your study can kind of translate to translational, right?
That's the exciting part. Yeah, it is. Yeah, thank you. Yeah.
Can I ask a quick question, please?
Yes, please.
Can I ask a quick question?
Yeah.
Oh yeah. Thank you. Actually, I'm a neuroscientist. Actually, I was a postdoc in Dr. Putin's lab in Mount Sinai. When I neuroscience department, we were working on how smoking causes diabetes. The nicotine activates the habenula enjoy the semicircular activation to impair the insulin release from pancreas and leads to diabetes, increased diabetes risk. Actually, based on that project, we have developed some chemicals, and we found that can increase the beta cell mass. And we submitted this paper to Nature last year, but was rejected because the editor asked questions about us whether our chemical is better than GLP-1 therapies. And we found some models and showed that yes, in our genetic models it's better, but the editor doesn't like it. But anyhow, we're still working on that. My question is that because I really new in this field, I really can tell the difference between the like proliferation, and differentiations. Because at your mechanism, so there are two separate pathways. One is DYRK1A to drive the proliferation. The second is PKA dependent the differentiations. But when I read a paper a lot because I know I'm new to this field about the regeneration.
Thank you. Yeah.
One, I don't find that you do any studies in type 2 diabetes yet. Only type 1 diabetes. The first. The second, actually, there's papers show that GLP-1 they can drive proliferation by PKA mechanisms. So the two separate pathway maybe it's not separate pathway. Maybe there's like one common pathway to drive like the beta cell regeneration. Because are there two pathways really
separated, or they have maybe some common mechanisms there.
So you mean that these two pathways could be together driven? What's
Yes.
your question? Yeah, I mean,
Because
there's
actually, when you mentioned that okay, Harman can act indirectly to activate the PKA pathway, CM, the
right.
PKA CREP pathway for the differentiation, right? And actually,
Right.
there's some literature showing that this pathway can drive proliferations.
Yeah,
So
I mean,
my question is that yeah.
The PKA actually, the pathway driven proliferation only in the rodent, but never happen in the human. Rodent cell line, you do see some of that proliferation, but we test that in human, you cannot driven the human base cell. Human base cell proliferation much more resistant than the rodent. It's a very different mechanism. So, yeah.
Oh, I see. And the second one is about actually all the other people have asked these questions. So at this moment for the clinical trial, are you going to target the type 1 diabetes? The major
you mean
type 1 diabetes?
You mean R12?
The patient. Yes.
You mean the patent? What is you asked that? What's the question?
For example, you're learning
the patient that you recruit for your clinical trial, is that type 1?
I see. Sorry. All right. So the patients, we know all the phase 1 is normal. No, not patient. It's all normal for that stage. Yeah. What I showed. Yeah. But eventually for the patient will be the phase 2 and phase 3. So that will include the real patients. Right now, no. Yeah.
Okay. Yes, because when I learned this field, I read some literature, but I don't have really expert in this field. But actually, in my Zoom, actually, in my friend Zoom, and I found that in just one of the PI mentioned in the late stage of type 2 diabetes, your beta cell loss. And then they say that you can have this regeneration by 3 mechanisms. One is the proliferation, second one is neurogenesis, third is the chance differentiation from alpha cell to beta cells.
Right. I mean, that's people generally.
It's a hypothesis. It's not. But
I mean,
it's
it's not just hypothesis. It's happening. It is happening. Every people. Yeah, it's all this pathway. There are people work on that. Indeed, it's happening. So yeah.
But if Harman one day will in the late stage animal models, seems like these two your mechanism are opposite each other. One is proliferation because you don't want the beta cell to get more differentiation, right? You want to get more chance differentiation from alpha cell or other delta cells to beta cells. Do you think these two pathways may be not in the same direction?
No, we try to get more beta cell with better function.
Target the beta cells.
Yeah, beta cell with better function. It's enhanced beta function.
You mean that too?
I'm saying that you produce more beta cell, you gain more beta cells, and also with better function. Same beta cell, some of it is not better good function. So that's right now for those things study. So
more beta cells and more better functions. Okay.
Right.
Yeah.
Right.
And I have one last question. Actually, I know that there's Harman, also there's Harman, no Harman. They all actually enrich in the tobacco leaves. I'm not sure whether there's the Harman similar as no Harman or Harman.
Well, I do notice those kind of things when I first find Harman. We do test all of this. Harman is the only one have the effect. All the other no. The answer. I do know those things. So I mean, you can see my original paper. We do compare several different Harman. Those different based on the 2D structure. It's almost identical. Just change a bond, but the function structure completely change different for 3D. So yeah.
Thank you very much. There's more questions.
You're welcome. Yeah.
Yeah. This is great. This is really intense discussion. This is really inspiring. Talk. So
all these questions also inspired me to think about.
Yeah.
following Xu's questions about the background of this DEC1A function. Since this inhibition of this pathway can increase beta cell regeneration, right? So whether it also needs to be inhibited during beta cell compensation in response to glucose increase, no, because during compensation stage, beta cell mass is also increased, right?
Can you repeat the question?
Yeah, the beta cell compensation stage. So the beta cell mass increase, whether this DYC1A needs to be inhibited to allow beta cell expansion.
Right. Yeah. I mean, whether DYC1A inhibition is required for increased beta cell mass, right?
Yes, yes.
Yeah, that's what we are, doing. That's we have to inhibit the DYC1A now.
Okay.
Yeah.
Yeah, so in other stages for during beta expansion, for example, in pregnancy people, late pregnancy moms, they also need to expand the beta cell expansion needs to, the beta cell mass needs to be increased to deal with the insulin resistance during late pregnancy. And for obese people, insulin resistance, all this cause beta cell expansion, right? So during this stage, this DYC1A needs to be this molecule decreased or function be inhibited?
Well, I really don't know those clear answer because we really don't compare those things situation. Really hard to answer.
Okay, yeah, probably
Right.
more research
Right.
to see whether it is a mechanism there.
Right. But we yeah, I never test those. I cannot really give you the clear answer for those things. So yeah.
This could be mutable PIR1.
Right.
Yeah, another question is about the Xu's question regarding the function of this beta cell induced by Howling. So humming increased beta mass. Have you ever tested the responsiveness of the beta cells to glucose challenge to see whether the insulin secretion still stay within the range? I mean, just measure the insulin.
We only test the whole, after treatment, the whole islet. We do see that enhance the insulin secretion, but will not be able to test the increase the new base cell because it's a very small population. We have to find a way to trace those cells. So we don't.
Yeah.
You know what I mean? It's a whole the islets treated with compound. We do see there's some increase
but the whole function but the whole mass within the range, together with all the other cells that function response to insulin, that function you have already tested, right?
Right, we do that. We have that increase the secretion with treatment. But term to the new increase the base cell, we could not do that at this moment.
I remember that question. Yeah.
Okay, I think it's pretty late right now. We definitely should let Dr. Wang have some rest.
Thank you.
Since he's still recovering. Well, anyway, I feel like this is really informative talk, and thanks Dr. Wang again, and also for all the panelists for joining us today. And we also thank all the audience for you to be here. And I think that's all for today's section. We're looking to see you guys again in our next section. Yeah, thanks you all. Have a good weekend. Have a good night.
Thank you.
Okay, thank you.
Thank you very much.
Thanks,
bye.
Roger.
Okay, bye.
Thank you.
Bye.
Yeah.